RNA polymerase 1 and RNA polymerase 3, they make uncapped RNAs. They don't have all of this, right? And that's why they don't have a tail, right? Or a cap. Because of that, they are slightly different than the mature mRNA. Okay, now, why do we do this? Why are we adding a cap? Well, the cap is important for mRNA because it bonds to the cap bonding complex in the nucleus. Okay, and that cap bonding complex is so important because it allows the mRNA to be processed and exported, right? Okay, now, the mRNA that's produced prior to splicing, like I said, is the pre-mRNA. And what we're doing right now, we add the cap and we're processing the mRNA. We're going to splice it and add our tail, and then we will have a mature mRNA. Okay, so when we go to splice it after we've added the cap, each one of these splicing events is going to remove one intron, and it's going to connect to adjacent exons. And when we connect them together, that's called a transesterification. Okay, a transesterification. And it's going to remove the intron as a little loop, like a lasso. Each one of those little introns, you cut both ends of it, and the ends fold up on themselves, and they make a little lasso or a lariat. It's called a lariat. Okay. Now, there are definitely advantages to doing this RNA splicing. Okay. For example, 60% of the human genes, these pre-mRNA transcripts, they can be spliced in a variety of different ways. So, in some mRNAs, you cut out an intron, but maybe in another mRNA, with the same sequence of exons, introns, exons, introns, exons, introns, you don't cut out the first intron. Maybe you cut out the third intron. Or maybe in another one you cut out both the first and the third intron. So the point is, the end result is that you end up with multiple mRNAs made from the same pre-mRNA, right? All by just cutting out different versions, different versions of cutting of the introns and then gluing the exons back together. Okay, so these introns too, the other thing about them is that they're not all the same. They range in size from 10 nucleotides to about 100,000 nucleotides long. Okay, and these exons and introns, how does this splicing, how does it know where an exon ends and an intron begins? Well, the way that it knows is that the borders between an exon and an intron are marked by consensus sequences. Okay, so it's looking for, just like in a promoter, it's looking for those specific, nucleotide orderings so that it can stick to it and know, hey, this is the difference between an exon and an intron, right? Sitting right on the fence between the two. Okay, now the splicing is performed by RNA. That's right. Well, at least it plays a huge role, right? But remember, the type of RNA I'm talking about is the small nuclear RNA, or snRNA. Little s, little n, RNA, right? And that RNA is about 200 nucleotides long, okay? And there's a huge, complicated scenario that unfolds in order to do the splicing. But basically, it involves several different RNAs, snRNAs. They're called U1, U2, U4, U5, and U6. And just to quickly and briefly go through it, but I don't necessarily think that this is something that we need to, focus all of our energy on. But each of these little snRNAs, it actually complexes with protein subunits. And the protein plus the snRNA, those U1s to U6s, right? They make up something called an snRNP, or a SNRP, right? A little tiny SNRP. And that's a small nuclear ribonucleoprotein. Okay, small nuclear ribonucleoprotein. And these SNRPs, well, they all come together and they form a core of a big thing called the spliceosome, right? And the spliceosome is what carries out the splicing reaction, okay? There are several steps to how this works, like, the branch point between the exon and the intron. It's recognized by a branch point binding protein, or a BBP. And then, one of these SNRPs, U2AF, which is a helper protein, it sticks to the branch point, okay? And then the U2 SNRP displaces the BBP and the U2AF, and then it base pairs with the consensus sequence between the intron and the exon. And then, a U1 SNRP comes in and base pairs with the 5' splice site. And then a U4, U6, and U5 triplet of these SNRPs comes into the spliceosome. And then the U4 SNRP gets bumped out. It gets ejected. It's gone. And then the U6 comes in and it displaces the U1 at the 5' site. And that cuts out the little lariat and the intron is gone. Like I say, it's a little complicated. We're not going to worry so much about it. But anyway, if that's how it happens, in case you wanted to know. Okay. But one thing we do need to think about, how do we choose the proper splice sites? How do we know when we've chosen the proper site? Okay. Well, don't forget the spliceosome, this big spliceosome made of all these SNRPs. The spliceosome is associated with RNA polymerase II with the tail. Okay, so the spliceosome is going to see the 3' end first as it's growing out, and it's going to allow the spliceosome to keep track of each exon and each intron as it grows from the tunnel of the RNA polymerase. Okay, now there's a working hypothesis called the exon definition hypothesis, because really we don't know everything that happens at this point. We don't fully understand this process. Okay, but basically the spliceosome components, they're called SR proteins because they're rich in serines and arginines. These proteins in the spliceosome, they assemble onto the exon sequences, and they mark off the 3' and the 5' splice sites, and they stay stuck to the growing mRNA as it emerges from the RNA polymerase. Okay, and then those SR proteins, they're going to work with the U1 snRNAs that I mentioned at one end, and the U2AF proteins at the other end, and this allows the cells to recognize the splice sites, and then they can differentially splice the same transcript in many different ways. But how do they choose to do this? They don't have a brain. How do they decide which version of splicing takes place so that they make certain mRNAs and not others from the pre-mRNA? Really, at this point, it's just not understood. Nobody really can tell us at this point how the decision of splicing takes place, right? So maybe you could be the one to help us figure that out. Okay, but I will say, I'm just going to leave the splicing in a second, but I will say sometimes splicing can get a little weird, right? There's this phenomenon called trans-splicing, and a few karyotes can do this. For example, there are these parasites called trypanosomes. They're single-celled eukaryotes. They're protozoans. And they cause African sleeping sickness, the trypanosomes do. They carry out this strange trans-splicing. And nematode worms can do it as well. But basically what happens is that the exons from separate mRNAs get spliced together. They get glued together to form mature RNAs. So it's not like they're just taking exons from one pre-mRNA, cutting the introns out, and then gluing the exons back together. Instead, they're taking exons from two separate pre-mRNAs, cutting those exons out, cutting the introns out, leaving the exons, and then splicing the exons from the separate pre-mRNAs together. It's pretty wild. And who knows what drives the mechanism to do that or the reasoning behind it. So as you can see, this field of biology has a lot of growing to do. But anyway, at this point, let's take a quick break before we get to the 3' end of the mRNA.
Love is policy, y'all.